Hello and welcome to this video. We are Group 3 from STM19. We will discuss about Krebs cycle. Krebs cycle is also known as the tricarboxylic acid cycle, and it is a biochemical pathway that is used to generate energy to the oxidation of acetyl-CoA. It is also used for the synthesis of NADH, or nicotinamide adenine dinucleotide, and for the production of amino acids. Krebs cycle takes place in the mitochondria of the eukaryotes and the cytosol of the prokaryotes. The Krebs cycle uses the two molecules of pyruvic acid formed in glycolysis and yields high-energy molecules of NADH and flavin adenine dinucleotide or FADH2, as well as some ATP or adenosine triphosphate. Unfamiliar terms. Glycolysis. It is the breakdown of glucose by enzymes, releasing energy and pyruvic acid. It is primarily the first step in the cell respiration cycle. Carbon compound. Chemical substances containing carbon. More compounds of carbon exist than any other chemical element except for hydrogen. Oxidation. In an oxidation reduction or redox reaction, one atom or compound will steal electrons from another atom or compound. Catalyze. Cause an action or process to begin. The first step involved is actually a preparatory step which begins with a pyruvate. Pyruvate is derived through the process called glycolysis of glucose, which is a 6-carbon compound, and it is split into two molecules of pyruvate, which is a 3-carbon compound. The next step includes the oxidation of pyruvate into acetyl coenzyme A by the enzyme pyruvate dehydrogenase complex, and in this reaction, a molecule of carbon dioxide and a molecule of NADH is generated. The acetyl-CoA is a 2-carbon compound. In the next step, the acetyl-CoA combines with OXALOACETATE, which is a 4-carbon compound, to form citrate, and hence the resulting molecule is a 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase. In the next step, the citrate is isomerized into isocitrate by the enzyme aconidase. In chemistry, isomerization is the process by which one molecule is transformed into another molecule, which has exactly the same atoms, but the atoms have a different arrangement. The next step involves the isocitrate being oxidized into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase, and in this reaction, a molecule of NAD is reduced to NADH, and a molecule of carbon dioxide is generated. Hence, the alpha-ketoglutarate is a 5-carbon compound since one molecule of carbon is lost into carbon dioxide. Next, the alpha-ketoglutarate is converted into succinyl-CoA by the enzyme alpha-ketoglutarate dehydrogenase, and in this reaction, also a molecule of NAD is reduced to NADH, and a molecule of carbon dioxide is released. Hence, the succinyl-CoA is a 4-carbon compound. In the next step, the succinyl-CoA is converted into succinate by the enzyme succinyl-CoA synthase. In this reaction, a molecule of GTP is generated. Next, succinate is converted into fumarate by the enzyme succinate dehydrogenase. In this reaction, a molecule of QH2 is generated, which is used for the production of FADH2. Fumarate is then converted into malate by the enzyme fumarase. And in the last step, the malate is converted into oxaloacetate by the enzyme malate dehydrogenase, and in this reaction, also NAD is reduced to NADH. Now let's look at the results of the Krebs cycle. Through each cycle of the Krebs cycle, following products are generated 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of carbon dioxide. Now, since glucose is split into two pyruvate compounds for each molecule of glucose, this cycle runs twice. Hence, the products formed are 6NADH, 2FADH2, 2GTP, and 4 carbon dioxide. All the NADH and FADH2 are next fed to the electron transfer chain for the generation of. So this is the Krebs cycle. Thank you for watching.